Hello, today I'll be discussing about receptors, ligands, and various types of receptors present on the surface and inside the nucleus. Before jumping into the topics receptors, I would like to discuss what are ligands. So ligands can be any drug, neurotransmitter, any hormone, toxin, or chemical substance which binds to the receptors and response is elicited somehow that we'll discuss in further videos. So now coming back to the receptors are the macromolecules and they are called the sensing devices. They take information and then transmit through the cell. Here I am drawing the structure of a ligand and its basic parts. You must all know. It has a ligand binding domain where the ligand is bound and an effector site whose conformation is changed when any ligand is bound to the ligand binding site. And when the conformational changes occur in the effector site, that eventually leads to a response. So the action of drug is only elicited, the response is only elicited when drug binds to something. But here's the thing, the drug is binding to plasma protein and showing no action. That means drug only show action when it binds to the receptor. So this is the concept. Now, have you ever thought of that why some receptors are present on the nucleus and others are present on the surface of the cell? Have you ever thought of that concept? Yes, the answer to this question is it's all about ligands. The story revolves around the ligands. It totally depends upon the nature of the ligand, their physicochemical properties that defines where their receptor should be. So taking the examples, suppose there are two ligands A and B. Ligand A is lipid soluble and can easily pass through the cell membrane. And ligand B is highly electronegative and large molecular weight compound. So it cannot penetrate the cell. So when it cannot penetrate the cell, the receptors are present on the surface of the cell. And for ligand A, the receptors are present inside the nucleus because it's lipid soluble. Coming to the examples of the ligands whose receptors are present on the surface of the cell, there are insulin, prolactin, FSH, which belongs to the category of hormones, unlike acetylcholine, noradrenaline, adrenaline, whose receptors are present on the surface. Now, the different types of receptors present on the surface can be categorized into three. The first one is G protein coupled receptors or 7-pass receptors, we call them, or 1-pass receptors or enzyme linked receptor, the second, and the third one is ion channels. Explaining G protein coupled receptors, they are also known as serpentine receptors. And why they are known so? Because of their shape. If we draw a G protein coupled, suppose this is a cell, and here drawing the G protein coupled receptors, the shape of the receptor is such that it looks like a snake. And because the cytoplasmic loops are going in and out of the cell 7 times, that's why it is known as 7-pass receptors. So showing the ligand binding domain and the effector site as well. It is also known as metabotropic type of receptors. So in short, there are various synonyms of these receptors. They are serpentine receptors, 7-pass receptors, G protein coupled receptors. So don't be confused. They all are same. Now coming to the next type of receptors, that is 1-pass receptor or enzyme linked receptors. As name suggests, the 1-pass receptors, that means the cytoplasmic loop only goes into the cell only one time. That's why they are known as the 1-pass receptor. And mostly, yes, they have the ligand binding domain and the effector site. Type of receptors are the ion channels. These are the gates present on the cell that open only to the particular stimulus, that opens only when a particular stimulus arises or comes. That can be a voltage or a ligand. That's why you have heard about the ligand gated ion channels and the voltage gated ion channels. So here they are. This is the end of the first video. These all are the topics which we covered so far. This was a very brief introduction on various types of receptors. In further videos, we'll take each receptor separately and we'll discuss them thoroughly. And yeah, thank you so much for watching.